Hello students, today's class and topic is electron transport chain. About electron transport chain, a sequence of electron carriers in the inner mitochondrial membrane are organized as electron transport chain. Whereby electrons are used to reduce molecular oxygen to water. During this process, large amount of free energy liberated, which is trapped in the form of ATP. So, what is electron transport chain? A series of electron carriers in the inner mitochondrial membrane. Electron transport chain, also called respiratory chain, entire sequence of events involving oxidation of foodstuffs and finally adding energy is known as biological oxidation. Now redox couples. Oxidation is defined as loss of electrons. Redox couples. Oxidation is defined as loss of electrons and reduction as the gain of electrons. Electron lost in oxidation is taken up by acceptor, which is said to be reduced. Oxidation reduction is a closely coupled process. When a substance is oxidized, other substance received means when a substance exists both in reduced state and in oxidized state, it is called an oxidation reduction pair or redox pair. That is given in example and in diagram. In this example, there are two redox pairs, NADH and NAD plus, and FMN and FMNH2. Different redox couples differ in their tendency to lose or gain electrons. Now redox potential. In oxidation reduction reactions, the free energy change is proportionate to tendency of redox pair to lose or gain electrons. This tendency can be quantitatively measured in terms of oxidation reduction potential or simply redox potential. At a standard condition, that is pH 7 and 25 degree centigrade temperature, that is redox potential. When a substance has lesser affinity of electrons, when a substance has lesser affinity for electrons than hydrogen, said to have a negative redox potential. On the other hand, If substance has more affinity for electrons than hydrogen, said to have positive redox potential. Thus, NADH, which is a strong reducing molecule, has a negative redox potential about minus 0.32, whereas oxygen, a potent oxidant, has a positive redox potential about plus 0.82. Electron flow from a redox pair with a more negative to another redox couple with a more positive. Redox potential of some biologically important redox system here given in diagram. More negative, hydrogen, NAD plus, NADH, NADP, FMN, FAD, pyruvate, lactate, auxaloacetate malate, pyrosuccinate, fumigate, cytochrome, and ultimately oxygen. So electron flows from electron flow occurs from redox pair with more negative redox potential to more positive redox potential. Now, component of electron transport chain or respiratory chain. First is the diagram of electron transport chain. This is diagram of electron transport chain. This diagram is including flow of electron. Various components also seen. Various complex in this diagram. Complex 1, complex 2, 3, 4 and 5. And various complex has its coenzymes as components. FMNH2, FADH2, cytochromes, NADH. These are components of electron transport chain. So, components of respiratory chain or electron transport chain. Nicotinamide nucleotide. First, nicotinamide nucleotide. NAD plus and NADP plus are two coenzymes derived from the vitamin niacin. Though they are formed from the same source, their functions are quite different. NAD plus is associated with catabolism and NAD plus is associated with anabolism. NAD plus is reduced to NADH plus H plus by dehydrogenase of different catabolic pathway. The substrate that are acted upon by NAD plus linked dehydrogenase include glyceraldehyde 3-phosphate of glycolysis, pyruvate link reaction, isocitrate, alpha ketoglutarate and malate of Krebs cycle, and beta hydroxyacyl CoA or beta oxidation of fatty acids. These substrates are acted upon by Na-linked dehydrogenase. For example, glyceraldehyde 3-phosphate substrate acted upon by glyceraldehyde 3-phosphate dehydrogenase. NADH is directly linked to respiratory chain. On the other hand, NADPH is not linked to respiratory chain but supplies electron to anabolic reactions such as fatty acid synthesis and cholesterol synthesis. So this was about nicotinamide nucleotides, NAD plus and NADP plus. Now flavin nucleotide. FMN and FAD are two coenzymes derived from the vitamin riboflavin. In their reduced form, they serve as component of respiratory chain, complex 1 and complex 1 and complex 2 respectively. Substrates that are acted upon by FAD-linked dehydrogenase include succinate and intermediate of Krebs cycle and acyl-CoA in beta oxidation of fatty acid. Unlike redox potential of NAD plus or NADP plus, redox potential of flavin nucleotide depends upon the protein with which they are associated. That is called flavoproteins. Flavoproteins can participate in either one or two electron transfer, hence they can act as bridge molecule between reactions in which two electrons are donated. Now third is iron sulfur proteins. Iron sulfur protein FES are constituent of respiratory chain complex 1, 2nd and 3rd. They occur either in oxidized form Fe plus 3 or reduced from Fe plus 2. There are about 8 FES proteins identified till date as a component of electron transport chain. All these FES proteins take part in single electron transfer, in which one atom of iron sulfur cluster is either oxidized or reduced. Now, fourth is coenzyme Q. Coenzyme Q, also called ubiquinone, since it is ubiquitous quinone in biological system. It is quinone derivative with a polyisoprenoid side chain. Structure is similar to vitamin E and K. Ubiquinone can accept one electron to form semiquinone or two electrons to form ubiquinol. Hence, like flavoprotein, can serve as bridge between two electron donors and one electron acceptor. Coenzyme Q exists in oxidized form under aerobic condition and reduced form under anaerobic condition. Now cytochromes. Cytochromes are proteins with heme as prosthetic group. Heme in turn is composed of porphyrin ring with an iron atom. Heme group of cytochrome is functionally different from that found in hemoglobin and myoglobin. Iron of heme in cytochrome oscillates between Fe plus 3 and that is in oxidized form and Fe plus 2 that is in reduced form, allowing them to function as effective electrons carrier. This is in contrast with iron that is found in hemoglobin and myoglobin, where exists only in ferrous form. There are three classes of cytochrome identified in electron transport chain, designated as cytochrome A, B, C, which differ in their light absorption spectra. Cytochrome B, C1 and C and A3 are the different cytochrome found in electron transport chain. Among these, cytochrome C is most mobile. Now, organization of electron transport chain. Inner mitochondrial membrane is constituted by 5 different enzyme complexes, denoted as complex 1, complex 2, 3, 4, and 5. Complex 1, complex 2, here is complex 1, complex 2, complex 3, complex 4, and 5. Complex 1 to 4 serve as carriers of electrons and are considered as components of electron transport chain or respiratory chain. These complexes are composed of flavoproteins, iron-sulfur clusters, hemes, and copper ions. These diverse transmembrane complexes contain multiple oxidation reduction centers. Complex 5. Complex 5 is an ATP synthesizing assembly of catalytic component known as ATP synthase. So, 1 to 4 serve as carriers of electron and complex 5 serve as ATP synthesizing unit. Apart from these, there are certain mobile electron carriers in electron transport chain which diffuse freely into the inner mitochondrial membrane. These include coenzyme Q, cytochrome C. Some even consider NADH and oxygen to be mobile carriers. Now, flow. Map for electron flow. How electron flow from different complexes. First, know how does electron transport chain operate. Electron transport chain consists of series of sequentially acting electron carriers. These components of respiratory chains are arranged in order of increasing redox potential. Hydrogen and electrons flow through the chain from component of the more negative potential, that is NADH, to component of more positive redox potential, that is towards oxygen or water. The electron flow within these transmembrane complexes leads to simultaneous movement of protons across the inner mitochondrial membrane. In other words, complexes first, third, and 4 acts as proton pump. Complex second does not act as proton pump. Only complex first, third, and 4 act as proton pump. Complex second does not act as proton pump. Now, electron flow from NADH to coenzyme Q. Electrons of NADH enter the respiratory chain at complex 1. Complex 1 contains about 46 different proteins including FMN containing phleboproteins and 6 iron sulfur proteins. Initially, the hydrogen and electrons are transferred to FMN, reducing it to FMNH2. From here, two high potential electrons move to iron sulfur proteins. Iron in the iron sulfur clusters cycle. Iron in iron sulfur clusters cycle between Fe plus 2 and Fe plus 3, that is in between reduced and oxidized state. Electron in the FES proteins are then moved to coenzyme Q. Entire shuttling process is coupled with an endergonic transfer of 4 protons from the matrix to the intermembrane space. So, the flow of 2 electrons from NADH to coenzyme Q, flow of 2 electrons from NADH to coenzyme Q leads to pumping of 4 hydrogen ions out of matrix. Now, flow of electron from succinate to coenzyme Q. Succinate dehydrogenase, the citric acid cycle enzyme, is part of complex second. It occurs as an integral membrane protein of inner mitochondrial membrane. Hydrogen atom pass from succinate to FADH2, then the electron pass through FES center to coenzyme Q. Leftover protons enter the matrix and are used to reduce molecular oxygen to water at a later stage. Complex second is much smaller than complex first, and in contrast to other complex, it does not pump proton out of matrix. Complex second never pumps protons out of matrix because the free energy change of catalyzed reaction is too small. Consequently, less ATP is produced from oxidation of FADH2 than from oxidation of NADH2. That is, from NADH2 2.58 ATP and FAD 1.5 ATP. Now, flow of electron from coenzyme Q to cytochrome C. Before this is Something about coenzyme Q. Coenzyme Q, also known as ubiquinone, as we seen earlier, because of its hydrophobic nature and its teeny size. It is fairly mobile within the mitochondrial membrane. Electrons are transferred from complex 1, 2nd and other sources to ubiquinone, forming semiquinone and finally ubiquinol, a reduced form of coenzyme Q. In accepting 2 electrons, coenzyme Q takes up 2 protons from matrix as it reduced to QH2. Accumulated pool of reduced coenzyme Q then reoxidized by complex third, pushing the electrons further in respiratory chain. Now transfer of electrons from coenzyme Q to cytochrome C. The path of electrons through complex third is complicated. This is because electrons have to transfer between 2 electron carriers, that is QH2, QH2 and 1 electron carrier cytochrome C. Finally, 2H2, that is ubiquinol, is oxidized to ubiquinone and 2 molecules of cytochrome is reduced. Flow of electrons through complex third is coupled with an endergonic transfer of 4 protons from matrix to intermembranous space. Now cytochrome C, second mobile carrier of electron transport chain. Cytochrome C is a water-soluble protein that occurs in intermembranous space. The single heme of cytochrome C accepts an electron from complex third and transfers it to complex 4. In complex 4, transfer of electron from cytochrome C to oxygen. Final component of respiratory chain called cytochrome oxidase carries electron from cytochrome C to molecular oxygen. Cytochrome oxidase contains 2 heme groups designated cytochrome A and A3 and 2 copper ions in addition to iron. After accepting the electron, the molecular oxygen combines with hydrogen atoms or hydrogen ions, that is protons, to form water. These protons, like the electrons, come from hydrogen containing coenzyme, that is NADH and FADH2. Only 2 protons are transported out of matrix per pair of electrons passing through complex 4 because of a smaller thermodynamic driving force. This is in contrast to the 4 protons each that are pumped out of matrix during flow of electrons through complex 1 and 3rd. So we are seeing there electron flow within these transmembrane complexes leads to simultaneous movement of protons across the inner mitochondrial membrane. And first, third, and fourth acts as a proton pump. First, third, and fourth complexes acts as proton pump. And complex second does not participate in proton pump. Electron flow start from NADH and is to oxygen by forming butter. Thank you. It is all about electron transport chain and flow of electrons. Thank you.